What is genomics? The first step to understanding genomics is to ask, what is a genome? A genome is the set of instructions needed to create an organism. Genomes are stored in cells, as DNA. DNA is short for deoxyribonucleic acid, an amazing molecule that stores information using a special code. You can think of a genome as a coded cookbook to build and grow an organism. For example, you. The human genome contains about 23,000 recipes that code for different proteins. Each recipe or functional unit of DNA is called a gene. But only about 2% codes for protein. The rest is non-coding DNA with regulatory or other yet unknown functions. The study of how DNA works is called genetics. The methodologies that define genomics allow us to see a bigger picture, a panoramic view of the entire DNA landscape. Not only how DNA works, but how the whole genome interacts with its environment, from the cell that houses it to factors outside the organism. The study of the genome and its environment is known as genomics. A famous example of the power of genomics is the Human Genome Project. The goal of the project was to read the entire human genome. That's enough information to fill not one, but 400 standard English dictionaries. As the project progressed, researchers needed to create new tools to analyze and share this massive amount of data. This, in turn, spurred an explosion of further research. By its publication in 2001, the Human Genome Project had forever changed the way we think about life sciences research. The speed of genomics. The Human Genome Project took 13 years and over $3 billion to complete. Since 2001, individuals have had their genomes read in a fraction of the time for a fraction of the cost. Soon, you'll be able to have your genome read quickly and cheaply. Expanding knowledge. Today, the genomes of thousands of different animals and plants and bugs have been read. By comparing genomes, we can see just how alike different species are, not only today, but through evolutionary history. Open access. The ever-growing list of freely available genomics resources is accessible to everyone, including you. Open access to such a wealth of knowledge allows many positive applications which impact our lives, our health, and our environment.
Another example of promising gene therapy involves muscular dystrophy. Muscular dystrophy is a genetic disorder characterized by insufficient production of a protein called dystrophin. Disease victims' muscles eventually weaken to the point where they cannot survive. Researchers studying dystrophy in rats have successfully used a harmless virus to transmit the missing gene to every muscle of the rat's body, reversing the muscle wasting that characterizes the disease. This is a magnified image of the quadriceps muscle of a normal mouse. The dystrophin protein is shown in green and outlines the cells in the muscle. This is a similar image of the mouse victimized by muscular dystrophy. And this remarkable image shows a muscle in that same mouse 6 weeks after the gene therapy. Protein therapy is similar, but instead of fixing a patient's genes, it is directed at artificially creating the correct form of the misshapen protein and injecting these correctly formed proteins directly into the patient. This may prove to be easier in the short term because we don't have to identify the specific genetic errors that cause the protein to be misshapen in the first place. We only have to supply the proteins that have the correct shape. Scientists have successfully produced a goat that has been genetically modified so that her milk contains a human protein that can be extracted and given to patients who cannot manufacture it themselves. On the not-so-wonderful side of this technology lie more disturbing potentials. We can now build viruses from their genomes. We have already been able to create the first synthetic polio virus. And this technique could be used to recreate terrible viruses from our past, like Ebola and the 1918 flu strand that killed up to 40 million people. What is even more worrisome is that there are easier ways of recreating microbes. You can simply add the right genes to a close relative. That raises fundamental questions about the wisdom of publishing the genomes of deadly pathogens. But in addition to curing diseases, we are also developing the skills to choose which of our genes our children will receive. Greg and Marie are about to hear the results of their in vitro fertilization from their genetic counselor. They are about to face the full implications of this emergent technology.
Your extracted eggs, Maria, have been genetically matched in vitro with Greg's sperm. Now, you've selected a boy with blue eyes, brown hair, and fair skin. As an adult, he'll be 5 feet 11 inches tall, and his IQ will be near genius level. Now, I've taken the liberty of eliminating a few undesirable traits: alcoholism, cancer, Alzheimer's, baldness, obesity.
Well, I thought certainly no diseases, but
we were wondering if we shouldn't leave some characteristics to chance.
You want to give your child the best possible start. Remember, this child is still you, simply the best of you. You could conceive naturally 1,000 times and still never get such a result. I'll give you a minute to think about it.
Should we choose the best that's in us for our children? If so, who should be involved in the definition of exactly what is meant by the best that is in us? If that scenario makes you uneasy, what about the next step?
Computer, access my personal medical file.
This woman is pregnant and is concerned about the features of her child.
Project a holographic image of the baby. Now, extrapolate what the child will look like at 12 years old. Display the fetus's genome. Modify the following genetic sequences. Extrapolate what the child will look like with those genetic changes.
If we give our children genes that we do not have, are they still our children?
Save changes.
Instead of selecting specific genes from each parent for our children, how about making identical copies of ourselves? Beings with identical genomes are called clones. Many plants naturally clone themselves in order to have offspring. Animal clones happen naturally as well. Identical twins have identical DNA and are natural clones. And now we can make a perfect copy of an adult animal. This is often a terrifically useful thing. Consider the goat mentioned earlier when we were talking about protein therapy. That goat could be cloned to form a flock of identical drug-making goats. In addition to fixing genes that are broken and choosing genes for the optimum result of an offspring, and even manufacturing new genes, we can now mix genes across species. We have already explored mouse genes in potatoes, cow genes in soy and sugarcane, and even human genes in corn, potatoes, and rice. We have put firefly genes in tobacco, and even put jellyfish genes into mice so that they glow under UV light. A little imagination might lead to people with gills who can breathe underwater, or people with sonar like bats or dolphins. Is Superman right around the corner? Like Pandora, we have opened the box, and it contains a huge amount of valuable information. We now hold the keys to understanding many diseases, and in the long run, curing those diseases. But there is other knowledge in that box that we must use to extreme caution. We will probably be very insecure knowing the information it contains for some time to come. But while it may make us uncomfortable, we should also find it both amazing and hopeful.